Now let's talk about the mechanism of glycogenesis. It is an important metabolic activity in which molecules of glucose in the body is converted to glycogen in order to be stored in the liver. Glycogen is synthesized from molecules of alpha-D-glucose. The process occurs in the cytosol and requires energy supplied by ATP and UTP. This is the overview of the mechanism of glycogenesis. It is composed of 6 steps which we will further discuss. First, glucose is phosphorylated into glucose 6-phosphate in the liver by the enzyme hexokinase. Second, glucose 6-phosphate is isomerized into glucose 1-phosphate by phosphoglucosamidase, passing through the obligatory intermediate glucose 1,6-biphosphate. Third, glucose 1-phosphate reacts with UTP to form the active nucleotide uridine diphosphate glucose. This is catalyzed by the enzyme UDP glucose pyrophosphorylase. Pyrophosphate is formed, which is later hydrolyzed by phosphosphatase into 2-phosphate molecules. Fourth, the UDP glucose will be attached to the glycogen primer. The process is catalyzed by glycogen synthase. More UDP glucose will follow the same process. That will lead to the elongation of alpha-1,4-glycogen chain. When the chain is at least 11 glucose residues long, the branching enzyme will transfer part of the chain at least 6 glucose residue to the neighboring chain to form an alpha-1,6 linkage, establishing a branching point. The glycogen grows further with the addition of alpha-1,4 linkages and branching points. It leads to the formation of a new non-reducing end. Besides the existing 1-glycogen chain will be elongated and branched. Now let's go to glycogenolysis, which is composed of 3 steps. Step 1. Shortening of chains. The enzyme glycogen phosphorylase cleaves alpha-1,4-glycosidic bonds between glucosyl residues of non-reducing ends until 4 glucosyl units remain on each chain before a branch point. The remaining structure is called limit dextrin, and phosphorylase cannot degrade it further. Step 2. Removal of branches. The branches are removed by the debranching enzymes. First, it removes the alter 3 of the 4 glucosyl residues attached at a branch. Next it transfer them to the non-reducing end of another chain, increasing its length. The remaining glucose residue attached at an alpha-1,6 linkage is still removed by the debranching enzyme, releasing it as free glucose. This glucosyl chain is not available again for degradation via the enzyme glycogen phosphorylase. Step 3. Conversion of glucose 1-phosphate to glucose 6-phosphate. Since the glucose released by glycogen phosphorylase is still in the glucose 1-phosphate form, it has to be converted first into glucose 6-phosphase via the enzyme phosphoglucosamidase. In the liver, glucose 6-phosphate is transported into the endoplasmic reticulum by glucose 6-phosphate translocase. Here, glucose 6-phosphate is converted to glucose via glucose 6-phosphatase. Hepatocyte release this glycogen-derived glucose into the blood to help maintain blood glucose levels. However, in the muscles, glucose 6-phosphate cannot be sent into the blood because of the lack in the enzyme glucose phosphatase. Instead, it enters glycolysis, providing the energy needed for muscle contraction.